I several times in the last month, because during the month I sort of think, what am I going to talk about on the next Friday meeting? And quite often I look for subjects that I don't know much about as well, so I can learn about it and then tell you guys about it. And a word that fascinated me for a while is something called metabolomics. It's quite a funny word to say. But I have a vague idea it's to do with assaying cellular activity. But I did not know one of the reasons it's being used is to develop it for use in more advanced diagnostics. So when I read this sort of thing, I thought that might be a great thing, a great subject to talk about. I don't know much about it, but we can learn about it together. And it has direct relevance to aspergillosis. So, starts off with putting things into context. What might the next 10 years bring? Today, I think I've mentioned several times, developments in laboratories, developments in medicine are just going so fast. Experiments I used to do when I worked in research would take me a month. You can probably do 100 in a couple of days now. It's phenomenal how things have got faster and things have improved. What I've ended up deciding to talk about is 3 developments that could change the way aspergillosis is diagnosed, could change how it's treated, and how it's monitored. And they're all pretty amazing when we scratch below the surface. It's the sort of thing I get quite excited about because I really am that geeky. Better diagnosis includes new biomarkers, and biomarkers is another word that I might go into in more detail another time. They're just things that patterns that scientists are collecting that if they detect in someone, it might be a specific hint that something is going on in their body, like, for example, aspergillosis. So a biomarker for aspergillosis will be absolutely amazing. We are developing them. We haven't got there yet, but they are coming. Metabolomics, I'll talk about in more detail shortly, but that is a fascinating thing to study and real sort of Star Trek-esque advance in diagnostics. And this work is going on right now. Artificial intelligence, we hear an awful lot about. It's a very important part of this, and I'll talk about that more shortly. Then we look at better treatments, and we're all aware of biologics. And if you were in the meeting on a patient's meeting on Thursday, one of us started talking about he's on omazilimab, which is Xolair for people who are familiar with biologics over the last 5-10 years. And he's been taken off Xolair and being given a different name of a drug. But what that is one of the first biosimilars. So just like a normal chemical drug, like anything like paracetamol, you've got the company that originally developed it, like any of the antifungals, and they're allowed, I think it's 20 or 25 years, exclusive access to the market. The only product you're allowed to buy is their product, in which case it would be voriconazole in that particular case. Other companies come along and are then allowed to copy it and manufacture it. In terms of chemical drugs, that just means reproducing the chemical, which is not easy. It's very difficult, but relatively straightforward science now. And they copy everything. And one of the benefits to patients, and the NHS in particular, is the chemical that they produce is going to be marketed without all the effort needed to do the research to discover the drug. They're just copying the existing drug. It's a lot cheaper, and you'll end up with a drug that's a fraction of the price of the original. Same thing's happening with biologics. Xolair is one of the first that we use. there is now a biosimilar. In fact, there are two. And this has been a long process because copying a biologic is not the same as copying a chemical. A biologic is significantly different to a chemical drug. I'll talk about that slightly more in a minute. We're still developing new antifungal medicines, so there's more of those coming on the market. I think we've talked a lot about resofungin, and there's I've forgotten the name now. Another one which is on the verge of coming into the clinic, so that's encouraging. But there are more coming on the way, which will further enhance the number of tools we have to treat fungal infections. And then there's something called personal medication or personalized medicine, which is been a name for quite some time now. But as we get more and more ways to precisely identify someone's personal health status, then more and more we're going to be able to design their treatment especially for them, which will have lots of advantages. Better decisions. This is what doctors do, make these decisions. But as we get more and more ways to diagnose, we get more and more test results. They can have hundreds of test results to analyze, and a doctor will maybe look at 2 or 3 of those to make a final decision after going through a process of looking at x-rays and asking you about your symptoms and so on. That job is getting beyond, in one sense, it's getting beyond the capacity of a human being to actually remember all this stuff and synthesize it into a diagnosis. But also, we are increasingly getting machines which are able to do this kind of analysis, and they can look at all 200 pieces of information, identify patterns, and come up with more consistent, hopefully safer, possibly even earlier diagnosis, which would be great. Home monitoring, a few people have mentioned now. One of the reasons the NHS is struggling a little bit is that there isn't enough capacity in hospitals. People are waiting for beds. Well, there's a very strong political move and a very, very strong movement by the NHS to start monitoring, essentially putting the patient's hospital bed in their homes, using lots of monitoring devices that the patient can use themselves. So all of your pulse and all of your blood oxygen, everything else that they need to monitor, they can do remotely across the internet. You're not left alone on your own, but you can manage. If you can manage on your home, you are presumed normally with someone with you, a member of your family, then this is a better way of looking after some patients, certainly not all of them, but some patients were better off looked after in their own home, and that's coming right now. We're starting to see virtualized wards where there are nurses sat here or wherever they sit to do this, monitoring 20 patients over the internet. It's pretty much the same job, except you need a car to get to all the places when you need to go there. More individualized care, that's pretty much the same as saying more personalized medicine. None of these will change overnight, but together they offer a glimpse of what the future may hold. This is the future of diagnostics and treatment in the NHS and in the world, really. Just to go through that in a slightly nicer. Do you like all the graphics that I'm drawing now, all by hand? The future of care, better diagnosis, there's metabolomics again, better treatments, biosimilars, better decisions, home monitoring and digital health, more individualized care. So we've got a flow of, there's your patient, and we take blood samples, scans, take symptoms, take wearables. All of this can produce things like wearables. Blood samples can now generate huge amounts of data beyond what a doctor is able to do. But we have AI, which is able to synthesize all of these things at the same time, recognize patterns, come up with patterns. All of that work has yet to be done, but there are example cases where this is possible already. The AI then comes up with a report, essentially, which the doctor then looks at and makes the decision about whether to what to do next. Usually an AI report would be saying something like, well, there's the signs of this and signs of this and signs of this. Well, quite strong signs of that and not very good signs of that. And the doctor would have to make a decision based on the analysis of the computer. As things develop, of course, AI is under massive development at the moment, then the AI will get better and better at pointing out these patterns to doctors. But the doctor will always be in there making that final decision. The AI can't communicate with patients. It can't care for patients. It's just crunching data, essentially. And if you have been here for some time, you know that we did with the university, we did 14 years ago, we did a series of doing genomic analysis of 100 patients. And that took years to declare its results, because when you analyze a genome of a patient, you're analyzing tens of millions of little bits of data, and it took a powerful computer to work out what was significant and what wasn't significant. Now we've got mega powerful computers that literally almost think for themselves in a way. That kind of work now is getting closer and closer to the clinic, simply because this is so reliable and so fast. AI won't forget anything, whereas we all know humans are fallible. And that leads to a very accurate description of what might be wrong with you, which also means that we can treat you early, we can treat you accurately, we can use the right drug at the right person at the right time, which is a series of phrases that you will be hearing over and over again. Again, this is the glimpse of the future. How am I doing for time? Oh my goodness, going too slow. Right, why are we talking about the future at all? How fast is medicine changing? Well, when many of us were first diagnosed, treatment options were much more limited. We had the choice between voriconazole and itraconazole. In fact, there wasn't any voriconazole till about 2010 or so. Biologic medicines weren't available. Genomic sequencing was expensive and slow, which I've just been talking about. Artificial intelligence didn't exist. Many of today's promised antifungal drugs hadn't been discovered. Now they're available for patients. Scientists couldn't measure thousands of molecules from a single blood sample. Now, this is a hint of what's coming, because this is metabolomics. Today, all of these are becoming reality. But obviously, it's not going to stop, and the next decade could bring even bigger changes. If we look at 2015, was when there were some new antifungal drugs. We had fungal culture, which is slow, often negative. We had things like CT scans, which were brilliant. Genetic testing was very slow. Travel forward 10 years, AI is beginning to help interpret scans. In fact, we already have AI in pretty serious trials here at MFT to look at X-rays for us as a screening tool to screen out normal X-rays from those that it raises a question mark and says to a doctor, you should look at this one. Biologic medicines are available for many patients. Biosimilars are starting to be used as of February this year. New antifungal drugs, new biomarkers, which are the things like galactomannan, which is a cell wall component from fungus that we can find in your blood if you've got an infection. And so same with glucan. And really amazingly fast sequencing tools that we have now. So we can actually, instead of waiting 5 years to interpret 100 genomes, we can now generate a genome in probably under a week and have the sequencing, the computer power to actually make that useful almost during a hospital visit, which is pretty amazing. Into the future, AI, metabolomics, which I'll go into in a little bit more detail, personalized medicine, home monitoring, and individualized medicine. It's coming. It's been slow getting here, but I believe this is a realistic time schedule. Why is diagnosing aspergillosis so difficult? I think we know mostly why that is. We breathe in these spores every day. The fungus is common. Many healthy people never become ill. Why is that? Some people become colonized but not infected. Others develop allergic disease, and so on. This is saying, so all those issues, and then we have to say, well, is it an allergic disease? Is it a colonization? And this has become an area of thought. Just because we can detect the organism in your airways, i.e. if you've got bronchiectasis, we can detect something like Pseudomonas or Aspergillus. It doesn't mean it's causing symptoms. It might just be sitting there quite innocently colonizing your airways, which is where they are normally found. And they may not be causing any problem, and they may be left well alone. Often no antifungal treatment is needed. Or infection, rapid. We can have rapid infection like acute invasive, or we can have chronic slow infection like CPA. But this is very distinctive from these other types of aspergillosis. There's no single test, so doctors will start with your symptoms, examine, take blood, give you a scan, take sputum sometimes. Here we can do a wide range of testing, so we test lung function as well. We look at your previous history, and this is becoming something we can do with great clarity now that we've got a huge computer system which tells us what your history was all in one place. And then we can make a diagnosis, or the doctor can make a diagnosis. Quite a long process using all these different tools, which are really good, but none of those tools is diagnostic on its own. If you've got high IgE, it doesn't mean you've got ABPA. You also have to have signs on your CT scan and probably some of the correct clinical symptoms and so on. It's a spectrum of symptoms. So then what your doctor does to make a diagnosis is like putting together a jigsaw. Your history, your lung function, your blood test, and so on. And this is telling us something, but none of these single tests tell us everything. Put them all together, and it enables the diagnosis. However, if we look deeper, so we've got, this is an iceberg. This is the level of the water. This is below the water. As you know, there's a lot more below the water than there is above the water. And what's above the water can sink ships. Most of the disease is happening long before it appears above the water. We can ask for symptoms. We can do blood tests, and that's about as much as we can do above the water. We are able to go below the water and go more deeply using imaging. But also, nowadays, we can look at protein, which is from tissue taken from the patient's body, probably mostly blood at the moment, I would have thought, or could be bowel, lung fluids and that type of thing. And we can look for all the proteins in that sample. But I'll go into more detail about that another time. We're not concentrating on that today. We're concentrating on something called metabolites. And metabolites are your body working to stay alive. We've got proteins, we've got DNA, we've got RNA, and all of those are incredibly important. But the things that are actually keeping you alive are much smaller elements, which are called metabolites. These are things that have been produced by your body functioning, by your body living, and they are highly complex. What metabolites are used for is they are telling us about what's happening in your body right now, today, this hour. It's immediate. If we look at DNA, It can tell you what might happen in the next year or two. If we look at proteins, it's relatively similar, but metabolites tell us about now. So metabolites show the chemical changes happening right now, often before symptoms. So looking at metabolites, we could probably make a preliminary diagnosis of, for example, ABPA or another form of aspergillosis before the symptoms are showing. And this is a dream for the future. It's a dream right now, but for the future, if we are able to do a blood test and look at your metabolites and how your body is working in real time, we might be able to make the diagnosis before you've even had any symptoms, which is amazing when you think about it. And that's why metabolomics is a study. It's part of this word, metabolomics, the analysis of metabolites. Metabolomics looks at thousands of tiny chemicals in your blood, known as metabolites. They're the intermediary between all your protein and your DNA and RNA working and keeping you alive. What they're doing is producing these metabolites. So if you are ill, maybe your metabolites will change. If you've got aspergillosis, maybe your metabolites will change because you've in a particular pattern because you've got aspergillosis. If we can detect what that is, then we can detect aspergillosis before the patient experiences any symptoms. This is the way that research is moving. We're going to try and get there before. So far, we're treating you after we've detected the infection, after the diagnosis is made. This is technology that we'll be able to go in before, and we're all aware of what that might mean. It's essentially the body's chemical fingerprint. Your body is carrying out millions of chemical reactions. These reactions produce tiny molecules called metabolites. They tell us what's happening inside your body right now. Unlike DNA or proteins, metabolites can actually tell us what is happening today and what is happening tomorrow. If we can predict aspergillosis, for example, and this applies to all types of disease that the human body experiences, if we can detect the right pattern of metabolites, we can predict a particular patient is going to develop ABPA, then we can treat that patient early, which makes a massive difference to the quality of life of that patient. It's a bit like if your body is a city, DNA is the street map telling you how the city is going to be laid out. Genes are the plans to make for buildings and high rises and all the rest of it, and homes. Proteins are the people that are constructing this city. Metabolites are the traffic zooming around in the city after it's been made. If there's an accident, the traffic changes, emergency service vehicles appear, congestion builds. You don't need to see this happening to know something is wrong. And the same is true inside us. And we can tell it from patterns that we can see in metabolites. So a healthy city, normal traffic flow, healthy person. City with an accident, traffic doesn't flow. All the traffic has stopped, it's acting abnormally, it's running out of petrol, people are getting angry. Changes happen in the car at the car level. Changes also happen in our bodies at the metabolite level. And that's what we're trying to detect. And all we need to measure these chemical traffic signals come in a single blood sample. So very simple as far as the patient's concerned, not quite so simple to actually make work. How does it work? Blood sample, laboratory measures thousands of metabolites, so it would go through a special machine to measure these metabolites. And that's half the problem. But we now have machines that can do this quite reliably. We also have massively capable computers which can analyze all these thousands of bits of data. And if you have 10,000 metabolites, the computer will be able to analyze your sample for particular patterns of metabolites, pick out a unique chemical fingerprint, which is your body working, and your doctors can interpret the results. This is the potential. This is what's being developed. It's not in the clinic yet, but certainly from the number of papers I've seen in the last year or two, this is a fast developing way we're going to be able to diagnose lots and lots of diseases. It's going to be, if it happens like we're speculating, then you can imagine this having as much impact as the biologics have on treatment. How do metabolomics work? It's like facial recognition. You don't recognize someone because you can see one eye or one ear or one nose. You recognize them because of the whole pattern, the face. And in metabolomics, work in exactly the same way. We see one metabolite, another metabolite, and another metabolite, and maybe 1,000 metabolites which are present or are working in particular ways that are all different from each other. Scientists aren't looking for one of these, they're looking for the pattern amongst thousands of molecules. One blood sample may contain up to 10,000 different metabolites. This is beyond the wit of any human to recognize those patterns. So until now, this would never have got any further, but now with modern computers, we can do this. I think we've gone through this enough, haven't we? Blah, blah, blah. Unique chemical fingerprint, doctors interpret the results, thousands and thousands of metabolites, use of AI, identify patterns related to diseases, and in our case, aspergillosis or vulnerability to aspergillosis. Thousands of metabolites change altogether. So we might envisage a healthy person. Each little dot represents a metabolite. And you can see there's a whole range of metabolites of different shades of green. But if you've got ABPA, maybe you've got yellow dots as well and red dots, where a normal person has green dots. So you've got a different pattern of metabolites because that's an allergic reaction. A CPA is not an allergic reaction, it's a chronic infection. So you're going to have different dots, different patterns, different colors coming up in your fingerprint, and the same with a bacterial infection. And these patterns are likely to be highly specific, may even tell us something about which bacteria is causing your infection way before we can now. It is amazing. Produces an overall pattern, an overall fingerprint of you with your body and your particular disease. And we use massive computers to generate these patterns. AI is capable of identifying these patterns and comparing patterns between a normal or between someone with CPA, and identify that person, as I say, for example, a 92% match with someone with CPA. And that might be someone who's going to develop CPA in the future. Who knows? This is the potential of new modern diagnostics, and this is starting to become quite close. Can it help aspergillosis? Probably yes. We haven't got there yet. We haven't been able to identify those patterns. That work is currently ongoing. Future applications, earlier diagnosis, which would be game-changing for aspergillosis. Being able to treat CPA before anybody has even got a cavity would be life-transforming, and we're hopeful that we can do that in the future. Better diagnosis. We can distinguish just by looking at this one pattern between an allergy, a colonization, or an active infection. So, yes, also you can monitor treatment. Is the treatment working? We can look in the metabolomics. We can predict flare-ups. So everyone who's currently saying, I'm feeling a bit grotty, I'm having a flare-up, well, we can actually predict them happening in the future and treat you with antibiotics before, a long time before we can now. Personalized medicine we've already covered. Current position, we got very encouraging research results. I may go into that in a bit more detail now I've introduced the subject another time. Most studies are still small. If you want to develop something into a routine part of the NHS in a clinic, it has to be done on large scale and proven. We've got reasonably good proof, but it's still very small scale, so we just need to scale it up right now. Larger studies are needed before it becomes a standard diagnostic test.
and that's the process by which that will happen. Yes, let's go on. So to conclude about metabolomics, it measures thousands of small molecules. It reflects what is happening right now in your body. And if you've got a blood sample taken now, the same blood sample could be completely different if you take it at 10 o'clock tonight. It is that sensitive. It looks for patterns, not one marker. It may improve diagnosis and monitoring, which would be amazing and life-changing for thousands of patients. But we're not quite yet there now. It's there at low levels, but we need to scale it up to bring it to clinics. Better treatment. Okay, so that's about diagnostics.